So today I'm going to be talking to you about combating bee bacterial diseases, specifically the advances in our diagnostics, as well as my work in the development of a vaccine. I would just like to firstly preface that there is some unpublished data in this presentation, so I will say that any slide with this icon here, if you could just not take any photos of that, that would be fantastic. So here's a brief outline of what I'm going to be talking to you about. So firstly, I'm going to discuss a pathogen prevalence study that was completed in our lab. Thank you. And then I'm going to go over a brief overview of AFB with the use of different diagnostic techniques that are available. And then I'm going to be going over the future prospects of AFB treatments with a particular focus in immunity and vaccine development. So a PhD student in our lab, Gopika Basi, has recently completed a study looking at the detection of common honeybee pathogens in honey samples. Note, this study is being published within the next few months, so you'll be able to get all this information and more when it is out. So just as an overview and an introduction to my work, I will just be presenting some of her results. So by extracting eDNA, otherwise known as environmental DNA, she subjected the honey samples to a PCR multiplex assay, which enabled her to screen for the below 7 pathogens. So in this study, a total of 135 honey samples from across Australia were tested over a 2-year period, being the 2023-2024 period. So all these samples were actually purchased for this study, with samples ranging from commercial to backyard beekeepers. So as the topic of my work is bacterial pathogens, I will just be briefly going over the bacterial results. So as you can see in the graph here, American and European foulbrood presented by the purple and grey coloured are prevalent across Australia, except for the Northern Territory and Kangaroo Island. Note, these results here aren't presenting the co-infections which actually exist within these 2 bacteria, but I unfortunately won't have time to talk about those today. So, however, this widespread presence of these bacterial disease is actually a frightening reality that the majority of our hives in Australia are affected by American or European foulbrood, and potentially both. So from these results, it's clear that an improved disease management is in need. But before I move on to that, I just really want to give you a brief overview of AFB and its significant difficulty for eradication. So to start, AFB's causative agent is Panabacillus larvae. It is a bacteria that is highly pathogenic, and it produces tough spores that make it really difficult to eradicate. These spores can survive in the environment for over 50 years, which is why the best course to stop the spread of disease is just the hive incineration. So on the right here is an overview of the AFB life cycle. So first, I really want to emphasise this transmission and dispersal of the AFB spores. Nurse bees are the main cause of transmission of the spores within the hive. So as they go around feeding the young larvae, they inadvertently transmit these spores around the hive. Once these spores are in the gut of the larvae, this is where they germinate, causing the active bacteria to occur. This is active bacteria then causes the outbreak. So these spores are the key reason to why AFB is so hard to control. They're tough, and as I mentioned, they stick around the hive environment, the honey, and ourselves for many years. Because of this, no treatment will work unless the spores are properly dealt with. Controlling these spores is critical to controlling the disease. So one treatment option that is available in the United States, but is actually prohibited in many countries, is the use of antibiotics such as oxytetracycline and thylacine. However, these antibiotics are actually ineffective against American foulbrood. This means that they don't actually eliminate the disease, they just mask the symptoms. Over time, research has actually shown that resistance to these antibiotics, making them even less effective. Additionally, there are concerns that these antibiotics can actually contaminate the honey, which is a major concern for us beekeepers. As a result, currently there is no true effective treatment for AFB.
Why would they use it in America?
Answered my question very nicely.
So they're like trucks.
I'm just going to keep my mouth shut. We do, in Australia, have a range of diagnostic techniques to identify these spores, one of which is the honey culture test, otherwise known as the HCT. So this test is currently the gold standard to identify AFB because it allows a quantitative level of infection to be identified. So quantitative means a level of detection, not just a yes, no. The other molecular techniques that are also used are PCR and LAMP. Both of these detect DNA at really low levels, however, they're not quantitative, so they are significantly more sensitive than our HCT, but they don't allow us to get a quantitative level of the bacteria detection. So this is super important. As I mentioned, the study that was previously done in our lab, that AFB is prevalent across Australia. But is this data telling us that there is currently a high level of outbreaks across Australia, or is this assay that was previously done, is that just detecting those subclinical levels of infection? So it's really important to understand the difference between clinical and subclinical levels of infection. So clinical levels of infection are those that are found on a HCT, while subclinical levels involve those spores being present in the hive. However, it's not at high enough concentrations to produce a positive HCT result. This is something I really want to emphasize to you all, is that spore presence within the hive does not necessarily equal clinical outbreak. One study has actually shown that hives with no noticeable signs of disease can actually still contain around 158 spores per bee. If we just put that into context for a second, that is a lot of spores. However, this same study found that even in a small increase of about 30 to 40%, a clinical outbreak of AFB occurred. So this relatively small increase really shows how easily the spore levels can tip the balance and lead to disease. So I was curious, is this eDNA detection that was found in the honey samples from Gopika's work, is this at the levels that are required to be positive on a HCT? Since HCT positive result is indicating that an outbreak is either occurring now or it's going to occur very soon. So I wanted to see how prevalent clinical infections were of AFB around Australia, because, as I mentioned, all of these samples were purchased and readily available on the market and therefore should be complying to AFB management strategies. So that's what I did. I used the HCT protocol on each of the PCR positive AFB honey samples from the previous study, and the result was pretty terrifying, that out of the 28 samples that tested positive for AFB on a PCR, 23 of those returned a positive result on HCT. So that means 82% of samples that tested positive via just spore detection on a PCR also detected positive on a HCT. So this is quite concerning and a clear indicator that either our current managements aren't being followed or they're just not working. So what are the outcomes of this work? Well, firstly, we have a new assay to detect multiple pathogens non-invasively, which is fantastic. 2, that there is a widespread presence of clinical AFB infections across the country. And 3, the current AFB management is insufficient. However, there are issues with changing our AFB management strategy. The first one is, in Australia, we already have some of the most stringent strategies for controlling AFB. 2, the current treatments that are available, but they are prohibited in most states, which is unfortunate, but these available treatments, whether they're banned or not, they're actually not effective. So what's next? What is the way forward? So that's where my work comes in, which is centred around immune priming, which is otherwise known as vaccination. So before I jump into vaccination and immune priming, I would just first like to introduce to you the honeybee immune system, which is really important to understand all of these concepts. So honeybees actually have a robust immune system, and it's comprised of a range of diverse mechanisms, all pictured here on the right. So these mechanisms all work together to defend against pathogens. The first are the primary defences, which are for bees, the exoskeleton and the periotrophic membranes. So these are just like the skin and hair on us, and they are the first line of defence to protect from pathogen entry. The secondary defences consist of cellular and humoral responses, which for us include our antibodies and inflammatory responses. However, bees lack this adaptive immune system and therefore don't produce antibodies. They do, however, produce a protein that is gaining a lot of attention for its role in passing the immunity down to its offspring. This protein is known as vitellogenin, or I'm just going to condense it down to VG because it's a bit of a hard one. So this protein actually acts to transport nutrients to developing eggs. So this protein actually provides the foundational basis of the AFB vaccine that is currently available in North America. So the vaccination of honeybees is possible through harnessing the mechanism of immune priming. So what's immune priming? Well, it's a memory-like response that helps invertebrates boost their immune responses against pathogens. So when an invertebrate is first exposed to a pathogen or pathogen-derived materials, its immune response. So this response is then remembered, or otherwise we call it primed, meaning that the immune system will then generate a faster and stronger response when the same or similar pathogen is then encountered. In honeybees, there is an extra phenomenon which immune priming can actually pass on to the next generation, which is known as transgenerational immune priming, which is otherwise shortened to TGIP. So the TGIP process is essential to understand how immunity is passed on to offspring. This mechanism is similar to how mothers pass immunity from their breast milk to their kin. So vitellogenin, or VG, and royal jelly are key players in this process. Investigations have shown that both of these have a role in influencing bees' immune response by regulating the production of antimicrobial peptides as well as enhancing the activity of immune cells. So looking at the image on the right here is the predicted mode of TGIP upon infection with a pathogen. So when the nurse bees infect the pathogen, the pathogen fragments are then incorporated into the royal jelly, as seen here, and then either fed to the young larvae or the queen. It is thought that these fragments then prime the immune system, providing future protection upon exposure. So the significance of this is, if we can trigger an immune response upon pathogen exposure, the spore load and therefore the production of infectious cells within the hive will reduce, therefore providing protection for the entire hive. So in the image here is a simplistic overview of the development of the Dellen Health vaccine. So this vaccine works by inactivating the AFB bacterium, which is then fed to the queen over the 8-day period. So inactivation is pretty much where causing the bacterium to stop replicating. So when the queen is fed this inactivated bacterium, it is then transported to her eggs via VG, which then primes her offspring. So this vaccine has an efficacy of around 30 to 50%. This doesn't seem like a lot. However, as I mentioned earlier, even a 30 to 40% spore load increase can trigger a clinical outbreak of AFB. So if we can reduce the larval death even by as little as 30%, it could potentially prevent an outbreak from manifesting. This finding really highlights the critical importance of managing the spore loads to prevent disease escalation, as a complete eradication of AFB is actually nearly impossible. Instead, our focus now really shifts to preventing these spore levels from reaching these clinical thresholds. So this has led me to my PhD work, which is based around a development of a single-dose vaccine against AFB as well as EFB. Because as we can see from the results I just presented earlier, that the current management is not effective and we urgently need new strategies for managing AFB infections. So as I mentioned before, my work Although my work uses the foundational basis of the Dellen Health vaccine, the concept and the design is actually very different. So our approach focuses on developing a bacterial component vaccine rather than a whole cell inactivated vaccine. So there are actually a few issues with having a whole cell vaccine, the first of which it requires the vaccine to be inactivated, which in this case is heat inactivation. So this method can actually damage critical immune sites, which have contributed to the lower efficacy of the vaccine because of the lowered immune response provided by the bees. The other major issue with this approach is that it's actually been found ineffective against other pathogens, where whole inactivated vaccines of various pathogens have been tested and provided no protection at all. So some of these pathogens include EFB as well as Israeli acute paralysis virus. So in contrast, our bacterial component vaccine is native and naturally non-infectious, and therefore it doesn't require this inactivation process to be completed. So this approach allows us to preserve key immune recognition structures which are crucial for triggering a strong immune response. By maintaining these intact structures, we aim to enhance the immune activation and provide improved protection against these target pathogens. Additionally, this strategy has a potential to serve as a carrier for immunity against other pathogens, including viruses, which I know is a hot topic right now with the transmission of Varroa. So I'm doing all of this work at AgroBio, which is a state-of-the-art science facility, but if we move away from the science for a moment, we've actually established an apiary that is less than a K away from our lab with over 30 active hives, which I've now actually recently learnt that it's one of Victoria's largest research apiaries, which we are actually continuing to expand. So this on-site research is managed by myself as well as Professor Travis, and here are some images of our current setup. I just would also like to shout out Ashton Edgley, who's been had a massive role in supporting us in this endeavour as well. So having this site really allows us to conduct practical and field-based research to combine our controlled lab experiments with real hive data to develop really targeted solutions for improving honeybee health, which we actually have a lot of projects in mind for, which is super exciting. So aside from the production of an AFB-EFB vaccine, my project will also be looking into the immune responses of honeybees upon exposure and vaccination. Alongside this, I will also be looking into the antibiotic-resistant strains of both AFB and EFB in Australia, as this is a major issue around the globe and there's currently no published data for this. Just this week, I've actually obtained some really interesting results in regards to the antibiotic strains of AFB and EFB, so if you want to chat to me about that later, feel free to. So for 2025, I have a few major goals. First was to establish our research apiary, which we did over the spring-summer period of 2024-2025. Then it was to isolate and characterise the bacterial component to use in the vaccine, which is really well under progress. So then when the season starts back up again in about August, September, we can conduct our safety trials to perform in vitro larval challenges post-vaccination. So that's really all I can say for now, as I can't delve too much into the specifics. However, I'm super keen to keep you all updated as I progress. So here are just some of my references. And before I finish up, if you are interested in this research, please feel free to reach out to me on the screen or scan the QR code. I am really actively seeking field samples, and any contributions would be really appreciated. Thank you. Feel free to take a photo. And are there any questions?